Dear students, today we will talk about gas chromatography. It is a technique used for separation of volatile compounds, and it can be also used for the separation of non-volatile compounds after derivatization. However, only volatile compounds are preferred and the slightly non-volatile compounds can be used after derivatization. I had talked about gas chromatography some time back, but I could not complete the topic, so I will start today from where I left it in my last class. So, a gas chromatograph has some components. Among the components, there is a carrier gas tank, there is a flow controller, there is a sample injector, there is a column, and the column is placed in a column oven, and then the column goes to a detector, and from the detector this signal is amplified and it is visualized on a monitor. So in this from this setup of a gas chromatograph, what we have understood is that a carrier gas is very much important, and this gas is the one which is used as a mobile phase in this instrument to carry along with it the sample which is injected through the injector, and uh, and when it enters the column, the entire length of the column helps in separating the compounds present in this sample, and these compounds get separated through its course along the length of the column, and when it comes out from the column, there is a spark which is induced by exposing the carrier gas to oxygen, and the spark is to ionize the compound which is coming out from the column, and and those ions are detected by this detector. So the detector which is suitable for this type of gas chromatography technique and detection of compounds is flame ionization detector, that is FID. There are other detectors also. The column is the heart of a gas chromatograph, and the column comes in different makes. For example, there are columns which are packed, there are columns which are capillary, there are columns which are super coated open tubular, and the carrier gas, there are three carrier gases which are commonly used: helium, nitrogen, argon, and also we can use hydrogen. However, hydrogen can be used only when there is no reactivity expected from this gas, that is hydrogen, with the compounds which we are interested in analyzing. So among the carrier gases, hydrogen has certain limitations, and the helium is quite costly, and nitrogen does not give good results, although nitrogen is used, but it is compared to other gases, it is not good. Hence there are even if it is costly, helium and argon, these are preferred gases. So the idea of gas chromatography column packing is can be understood in the way and that that thin layer chromatography plates, for example TLC plates, are made, and if one folds the column with the coated silica coated side inside, and the plate outside, then it is similar, it is just like a GC column, which is cylindrical, and inside the cylinder on the walls there is a coating of certain high boiling liquid such as silicone in its liquid phase, and it is mixed with diatomaceous earth, or there may be polyester liquid phase such as diethyl glycol succinate. These are different column materials: one liquid silicone in its liquid phase mixed with diatomaceous earth, and two polar polyester liquid phase such as DEGS about 15%. Among the detectors, flame ionization detector is one, thermal conductivity detector is another, and the photo ionization is also a type of detector. Although gas chromatography has many applications, it too has demerits. For example, gas chromatography can detect only volatile compounds or those which can be evaporated intact at elevated temperatures, or from which volatile derivatives can be reliably obtained. But only about 20% of non-organic compounds are volatile, so it cannot be used for the remaining organic compounds unless they are derivatized. But not all of them can be analyzed even if they are derivatized. Here in this slide, types of columns are shown, and among these, porous layer open tubular column plot is one, packed capillary column is another, and wall coated open tubular column, which is mentioned as super coated open tubular column in the previous slide, is shown. So these three types of columns are most preferred. Among these, the wall coated open tubular column is the one which is most commonly used. 80% of all the columns used in GC operations are with this wall coated open tubular column. So it has a dimension of 0.1 to 0.53 millimeters internal diameter, and the coated film on the wall of the column has a thickness of 0.1 to 8 micron. Now GC can be used for detection of compounds, as I said, volatile compounds, but identification of the volatile compound as well as quantification of those compounds is possible only when we have authentic reference standards. However, if we don't have authentic reference standards, we can use some detectors, special detectors, which can help us in identifying the nature of the compound eluting from the column. One such detector is mass spectrometer. A mass spectrometer measures the mass of the compound eluting from the column. The phenomenon or the process involved in measuring the mass of the eluting compound is almost the same as that of LCMS. Here in GC, when it is coupled with a mass spectrometer, it is called GCMS. So the mass spectrometer, what it does is, it here in this GCMS, the eluent from the column is directly entering the mass spectrometer chamber. There is no ionization before entering the mass spectrometer. Once it enters the mass spectrometer, there is a ion source which is in the mass spectrometer. It ionizes the compound which is entering the mass spectrometer chamber from the column. Once the compound or the molecule is ionized, it it since the mass spectrometer is maintained under vacuum, the ion which is which is generated by the help of ion source, when it releases ions and it hits the molecule entering the gas mass spectrometer, the ions are then focused to enter a section which is called ion trap mass analyzer. Here the ions are directed to ions are directed to hit a detector which is which is going to amplify the signal which is corresponding to the ion which is coming. This ion trap mass analyzer has alternating current, and because of that the ion uh, gets directed to this electron multiplier. Here when the ion hit, actually in this ion trap mass analyzer, the ions are separated. So if one compound entering from the column into the mass spectrometer and it is ionized, then its mass, based on its mass, the ion will be separated from the other ion. So each ion will be generating or giving out a signal which can be amplified with the help of this electron multiplier, and then it can be visualized on a computer. So in this way we will be able to detect a compound even when there is no authentic reference standard, but it is going to be detected based on its m by z value. M by z value is possible only when we use a mass spectrometry in tandem with a gas chromatography. So here in this slide there is one example shown of 2-nitrotoluene whose mass is 120 m by whose mass is 137, and it is being fragmented into its product ions, and these products ions have molecular mass of 120, 92, 77, 65, and like this we will be able to understand the identity of the compound which is eluting from the GC and entering the mass spectrometer. Now in contrast with gas chromatography, high performance liquid chromatography is another technique which is used for quantification as well as identification of compounds which are polar or non-polar in nature. A high performance liquid chromatography is nothing but a simple chromatography column which has been attached to a high pressure generating pump, and because of that the sample is separated under pressure, and this pressure is the high pressure is required to push the samples through the column. Liquid chromatography columns have very small internal diameter, for example 4.6 millimolar, 4.6 mm, and this 4.6 mm is full of compounds, full of matrix, and because of full of matrix it generates a lot of back pressure. So in order to counter that back pressure, a high pressure pump is required to push the sample through the column, and when it passes through when the sample passes through the column, different compounds are separated depending upon their polarity and depending upon their interaction with the packing material. And when it comes out of the column, different types of detectors can be used depending upon the nature of the compound to detect them. For example, UV visible detector is one such detector, which is nothing more than a detector that is used in a spectrophotometer. So it is quite like a spectrophotometer, except that the compounds are separated before they are detected by the UV visible detector. So here in a spectrophotometer, UV visible spectrophotometer, we get a collective signal, but here in the case of high performance liquid chromatography or HPLC, we will get peaks of individual compounds, because the compounds are being separated in the column, have been separated in the column. So there are other detectors also, but in the heart of this high performance liquid chromatography or HPLC is the column. The column has different chemistries. In the olden days, normal phase columns were used, in which the stationary phase is polar, such as silica, cyano, whereas nowadays reversed phase columns are being used, in which stationary phase is non-polar. So in the olden days, the stationary phase in the case of normal phase columns was polar, and now in the case of reversed phase, the stationary phase has become non-polar. So what changed this reversion or difference in the phase of stationary phases in normal and the reversed phase will bring about? One is that the mobile phase, which is used in the normal phase, is non-polar, whereas the mobile phase used in the case of reversed phase columns is polar, such as water, buffers, methanol, acetonitrile. In the case of normal phase chromatography, the elution pattern of compounds is uh, such that non-polar solutes elute faster than polar solutes, whereas in the case of reversed phase columns, polar solutes elute faster than non-polar solutes, which means that non-polar solutes will be retained for a longer period of time. Now there are many detectors. As I said, UV visible detector is one, and there is photodiode array detector. And here in this slide I have given a limit of detection of each detector. For example, a UV visible detector can detect up to 10 raised to power minus 10 gram per ml. A PDA can detect all 10 raised to power minus 10 gram per ml concentration of compounds, whereas a fluorescent detector can detect uh, up to 10 raised to the power minus 13 gram per ml. There is a refractive index detector which is commonly used for quantifying or detecting lipids, fatty acids, triglycerides, and other such compounds. In this RID refractive index detector, the limit of detection is up to 10 raised to power minus 7 gram per ml. Electrochemical detector is another detector wherein reactions can be reaction products can be quantified, and uh, uh, when there is redox type of reactions taking place, the quantification of such reaction products and the reaction itself can be done with the help of electrochemical detector, and its limit of detection is 10 raised to the power minus 9 gram per ml. In the in a HPLC or high performance liquid chromatography, a deuterium lamp or tungsten halogen lamp are used in order to generate a laser help in which will help in recording the spectrum of each compound or the absorbance of each compound in the case of UV visible detector. Here is a simple schematic diagram of an HPLC unit where a solvent reservoir is required, a transfer line with frit is required, and a pump with manometer is required. For in the line is sample injection, and the fifth component is a column with thermostat with provision for maintaining temperature, and 6 detector, and 7 in the uh, in the list of requirements is waste outlet where outlets can where waste uh, waste elute or a mobile phase which is eluted as a waste can be collected, and 9 data acquisition. This is how a HPLC chromatography system looks like. Okay, the mechanism of separation of compounds in a HPLC column is shown here. On the top left, what you see is the chemical structure of silica, which is the uh, matrix of HPLC columns. The matrix has silica which is sometimes derivatized or sometimes not derivatized depending upon the chemistry of the column for the end use, that is the particular application. For example, if the column is to be used for secondary metabolite quantification and or for amino acid or for triglycerides, depending upon that the chemical structure of the packing material is developed. One such a column which is commonly used these days is reversed phase C18, in which the silica here is derivatized with a C18 carbon 18 chain compound, and this derivatization renders the silica non-polar. Otherwise, silica is polar, and if we use silica alone in a column, then the column will be called normal phase column, and if we use a derivatized silica with octadecyl compound like a C18 compound attached to the hydroxyl group here, then it becomes a reversed phase column, and nowadays commercially reversed phase C18 columns are available. This is also called octadecyl silane because of the octadecyl moiety which is attached to the hydroxyl group of this silica. So when our compound here on the bottom right, what is shown is the compounds present in this sample when it enters the column, which is the stationary phase of this whole chromatography setup. The compounds are getting separated depending upon their interaction with the packing material, which is the stationary phase present in the column. So a few compounds will interact for a longer period of time, and other compounds will interact with the compound for less period of time. For example, in the case of reversed phase C18 columns, polar compounds will move faster. That means there will be no interaction with the packing material or the octadecyl silane, which is the derivatized form of silica, and because of that the polar compounds will elute faster, it will be detected faster, whereas the non-polar compounds, because of that hydrophobic interaction between non-polar compounds, they will be retained longer, and it will elute after all the polar compounds have got eluted. So this elution pattern is shown here in a chromatogram, which is taken from a published paper, wherein a sesamol, sesamine and sesamoline, which are compounds present in sesame seed oil. Here the sesamol with OH, which is polar, is eluted faster, and after this other compounds which are less polar are eluted. Here in this slide, another chromatogram, comparison of chromatograms is shown, in which different extracts like water extract, hexane extract, dimethyl ether dichloromethane extract, then ethyl acetate extract and the methanol extract are shown for a comparison. Here the polar in water extract, which is containing most of the polar compounds present in the sample, because water is polar, so it will be extracting only polar compounds from the sample. So polar compounds have early elution, they elute faster, and that is why if you see the extracts of water in water extract, you will see the compounds are eluting faster before 35 minutes. All of the compounds have got eluted, whereas hexane extract, which is completely non-polar, it will be able to extract only non-polar compounds. The elution of compounds starts only after 25 minutes of runtime, and it goes on up to 60 minutes, which means that the compounds which are present in this hexane extract are interacting with the column material for longer period of time, and because of that they are coming or eluting slowly, and it is taking time. So this difference, this time taken to elute from the column is called retention time. So non-polar compounds will have longer retention times in a reversed phase column, whereas polar compounds will have shorter retention times when they are eluted through a reversed phase column, C18 column. So similarly, other extracts can be also seen in this context. Now we have discussed two important methods of separation as well as detection of compounds. One that we have seen so far is gas chromatography, and then the another one is HPLC. The gas chromatography is used, as I said, for mainly separating non-volatile for separating volatile compounds, whereas HPLC is used for separating non-volatile or polar compounds. Gas chromatography is used for volatile or non-polar compounds. So what are the sensitivities and differences between these two techniques, these two important techniques? One difference among them is difficulty in separation. Gas chromatography can easily separate compounds. HPLC can also easily separate compounds. The speed of separation may be fast in both the cases. These are the points of comparison, and the instruments which we are comparing are shown here, and how they fare in the comparison is listed out here. So in terms of automation, gas chromatography can be automated, HPLC can be also automated. Adaptation of system to separation problem: gas chromatography, by changing the stationary phase, it can adapt to a new separation setup or separation system. And in the case of HPLC also, by changing the stationary and the mobile phase setup, we can adapt, or we can use it for a new separation problem. Another point of comparison is application of the scale of application or scope of application of these two instruments. GC has application only when the compounds are volatile and they are thermally decomposed, whereas in the case of HPLC the compounds should be soluble and these compounds should be um, in this compound, the compounds detected by HPLC will be limited only by their insolubility, whereas in the case of gas chromatography the lack of volatility and the thermal decomposability will only restrict its application in terms of separating or identification of the compounds. In terms of sensitivity, the points of comparison here shown are uh, the per column gas a gas chromatography column has around 20,000 number of separation plates. What is a separation plate? A separation plate is a theoretical concept wherein each compound or each a molecule can be separated from each other during the course of its during the course of its journey from the entry to the exit of a column. So so this is provided the theoretical plate or separation plate is provided by the interaction of the compound with the packing material. So in the case of a gas chromatography, the number of theoretical plates is uh, up to 50,000, whereas in the case of an HPLC the number of theoretical plates or separation plates is up to uh, if up to 50,000. So in terms of the sensitivity or in terms of the ability to separate compounds, these two instruments are uh, uh, equally capable of separating compounds. However, in a gas chromatography the columns used are quite long, so there will be more possibility of theoretical plates, because of which there will be better separation in a gas chromatograph. So among these two techniques, the sensitivity of gas chromatography is in general more than that of HPLC. We will stop here for today and I will continue this topic in the next class. Thank you for your patient hearing.